Hello friends, ബയോസിന്റെ പുതിയ വീഡിയോയിലേക്ക് ഏവർക്കും സ്വാഗതം. നമ്മുടെ കഴിഞ്ഞ വീഡിയോയിൽ നമ്മൾ ടൈപ്സ് ഓഫ് ക്രോമോസോം ആണ് നോക്കിയത്. Here we are going to discuss about the giant chromosomes or the Polyteen chromosome and the lampbrush chromosomes. Okay, so let's get started. So what are giant chromosomes? As the name suggests, giants are very large in size. Giants are very large. Okay, so these chromosomes are very large when compared to the normal chromosomes. Okay, especially enormously enlarged chromosomes. Okay, these are about 100 times thicker than the normal chromosomes, and these are of two types: Polyteen chromosome and lampbrush chromosomes. Okay, the name giant chromosome was given by Winchester for these chromosomes. Okay, so the first type of chromosome or giant chromosome is the Polyteen chromosomes. This was first observed by Balbiani in 1881 in diphtherin larva. Okay, in a diphtherin larva, and this is also known as salivary gland chromosomes. Okay, the name Polyteen chromosome was given by Kohler. Okay, and this is mainly seen in the interface of cell division. These are banded chromosomes and occur mainly in larval salivary glands, midgut epithelium, rectum, and malphizian tubules of various genera of diphtherians. Okay, this was first observed in the diphtherin larva by Balbiani. The name Polyteen chromosome was given by Kohler. It is also known as salivary gland chromosomes, and these are seen during the interphase of cell division. These are banded chromosomes and occurs in larval salivary glands, midgut epithelium, rectum, and malphizian tubules of various genera of dipterans. Okay. Now, the structure of the Polyteen chromosome. They are roughly cylindrical. Okay, you can see that these are cylindrical, right? These chromosomes are cylindrical. And the distinct pattern of transverse striated structures. These are cylindrical structures, and they have a striated structure. Okay, the banded regions and the interbands. Okay, darkly stained bands. These are the darkly stained bands, and these are the bands. Okay, while this is the lightly staining interbands. Okay, now the dark bands, you see here, the dark bands consist of DNA. They are rich in DNA with small amount of RNA and basic proteins, while the inner interbands contains less DNA and more of acidic protein, and because of this they are less active. Okay, so the banded regions are rich in DNA, and they have basic proteins and some of RNA, so they participate actively in the proteins transcription, because they have a lot of DNA. Okay, while the interface is mainly composed of less DNA and more acidic proteins, so that makes it less active in nature. Okay, now what do you mean by polytenization? These are known as Polyteen chromosomes, right? So what is polytenization or endoreplication? You can see that these chromosomes are not separated from each other. There are many chromosomes joined at a single point, right? This process is known as endoreplication. So these chromosomes are formed by repeated replication of DNA without division of chromosome to daughter chromosomes. Okay, these chromosomes are repeatedly they are replicating, but they are not getting separated from the parental chromosome. So they form a several loops like structures. Okay, this amplification without separation is known as polytenization. Another feature of this Polyteen chromosome is the presence of chromosomal puffs. These are the chromosomal puffs. Okay, these are the swelling near the centromere. Near the centromere, there you can see a puff-like structure, or these are also known as chromosome puffs, or and these puffs make ring-like structures in the Polyteen chromosomes, and these rings are known as Balbiani rings. Balbiani rings. Balbiani rings. The first person who observed the Polyteen chromosomes, Balbiani. Okay, and these rings or these ring area or the puff area are transcriptionally really active. This is the transcriptionally active region of the Polyteen chromosomes. Okay, what we have seen is Polyteen chromosomes are the giant chromosomes. Giant chromosomes, these are mainly seen in the larval of diphtherians. Okay, mainly in the salivary gland ducts, salivary glands of Drosophila. We study this in Drosophila. Next, the structure is somewhat cylindrical and they have striated structures with bands, darkly stained banded region and interband regions that are lightly stained. The banded region are rich in DNA and basic proteins, so they are active in the transcription, while interbands are rich in more acidic protein and less of the DNA, so they are inactive, less active. The other property of the Polyteen chromosome is the polytenization or endoreplication, where the chromosomal DNA replicates without division of the chromosomes into the daughter chromosomes, and this is known as polytenization. The other property is the presence of puffs or rings, also known as Balbiani rings, which are actively transcriptional area of the chromosome. Okay. So thank you for watching. Please share, subscribe, and like the video.